Hello, my name is Caleb Caicedo, an undergraduate student at FIU, Florida International University, and today I'll be discussing Dendrophylax lindenii, otherwise known as the ghost orchid. Dendrophylax lindenii is native to Florida and Cuba. It was first collected by John Jules Linden in 1844. However, it was first formally described as a species by John Linley in 1846. However, later its scientific name was then changed to Dendrophylax lindenii. Dendrophylax lindenii, or the ghost orchid, is currently listed as an endangered species by the U.S. Fish and Wildlife Service. This was due to its ornamental popularity, which led to it being poached from its native habitat. Aside from its ornamental value, the orchid has a high educational value through raising awareness about unique ecosystems and environmental threats. The ghost orchid is a leafless orchid whose body consists almost entirely on flattened photosynthetic roots. The roots contain a thick velamen and fluorescein layer that perform photosynthesis and help with water absorption. The stem is extremely reduced, only a few milliliters thick, and is hidden beneath the root mass. Large white flowers are adapted for hawk moth pollination with long nectar spurs and fused reproductive columns. Seeds are dust-like and depend entirely on mycorrhizal fungi for germination and nutritional uptake. These features enable its survival in the high humidity and low light swamp forest of Florida and Cuba. As mentioned earlier, this is an endangered species. So it goes without being said that there have been various attempts into conserving this species in its natural habitat. However, it's not as easy as it seems. For example, this species requires specific and constant humidity levels, an intact swamp canopy, and specific host trees such as popash and pineapple. So if these specific conditions are not met, then it's not going to do well when you reintegrate the species back into its natural habitat. Now, why would the habitat not have these requirements? Well, it's been a while, and as such, the habitat has gone through various challenges and changes. For example, habitat destruction and disturbances have limited the amount of these resources and requirements are present naturally due to hydrologic changes, hurricanes, canopy loss, and human activity. We've pretty much limited the amount of space and specific areas in the natural habitat of this orchid where it can do well and thrive. Additionally, this orchid has a low natural reproduction rate. It is rarely pollinated and relies heavily on a single long-tongued hawk moth. So this pollinator species must also be present in the habitat, and if it's not, then the integrated population from labs and other conservation efforts will not thrive long term because it's not being pollinated. Additionally, poaching pressure. Due to the highly targeted value this species has, it has been poached and removed. So if they're not being protected in this habitat in a long-term foreseeable future, then it's expected that poachers will end up removing them again from the environment. And reintroduction challenges. As mentioned earlier, lab-grown seedlings have a high mortality rate when they're being reintroduced into the natural habitats, and this is due to various factors. If the seedling was not properly prepped for for reintegration into the natural habitat, then it will undergo induced stress and end up dying without having a chance to reproduce. Now, it goes without being said that the conservation efforts done in protecting their natural habitats help tremendously. These protecting habitats such as Everglades National Park, Big Cypress National Preserve, and specific locations in Cuba help conserve not only the natural habitats and specific requirements these species' needs and their habitats, but also help protect any existing wild populations, which will help in the long run with genetic diversity. Now, that alone is not enough. As mentioned earlier, these species has a very low reproduction rate. As such, many institutions, agencies, and government funded programs have gone into actually trying to cultivate and propagate the species in a lab setting and to then reintroduce them into the natural habitat to the wild populations. However, that's quite difficult to do. This species is very finicky and sensitive, so developing a proper protocol is essential. And as such, uh, proper sterilization is extremely important. You must ensure that your sample size is protected from any fungal or bacterial diseases. As such, then having a proper culture medium is important. Many research um, that's being done currently use MS or half strength MS with sucrose and activated charcoal, peptone to really help the seedlings develop. And speaking of seedlings, most of the research that is being done in repropagating these species in vitro labs are using protocorms. Protocorms are a developmental stage of the orchid uh, right before it reaches a seedling phase. It's a very brief window in its growth, and it's very essential to use this stage to then propagate them properly in the lab setting. Another conservation approach is using protocone-like bodies. Now, what's the difference between using protocone-like bodies and just simply using protocorms? Well, protocorms results in genetically diverse individuals. It's essentially the natural way to do it in a lab setting. However, protocorm-like bodies are synthetically made. They're calyces that are made by inoculating an explant tissue with a specific PGR. The PGR is 2,4-D, right? And this is done following normal protocol, sterilization, and a culture medium that was used previously. However, you're adding this additional step where now you're inoculating a specific explant tissue, particularly in this case from the root shoots, to develop these calluses, which is seen in the image. This callus will then allow you to micropropagate in the callus into genetically identical individuals. It's coming from the same tissue, the same DNA essentially. Now, with all that being said, it all means nothing if the plants that are being grown in the lab, whether via protocorms or protocome-like bodies, are not able to successfully acclimatize themselves into the environment. This is where most of the research struggles when it comes to this species, as this is a very difficult achievement. The success rates are very low, and it involves removing the protocone-like bodies or seedlings from the culture, from the MS medium, rinsing it off gently to avoid any damage to the roots, to any tissue, and then you have to then mount on a host substrate, cork, bark, with sphagnum moss to maintain moisture, right? Place the mounts in a high humidity chamber with bright filtered light. You want to make sure that you don't burn the tissue of these delicate species that you just cultivated in a lab. And then gradually reduce the humidity for a time span of 4 to 6 weeks while increasing the ventilation. You want to prevent any rot, any fungus, bacteria from growing. Maintain the temperatures at about 24 to 28 degrees Celsius with a gentle airflow and daily misting. And then after 6 to 12 weeks, you want to transfer to a screen house or shade house with stable humidity, about 70%. Now, remember, this is a very delicate process and success rates are low. Many research that's being conducted struggle with this phase, this final phase of their protocols. And it's because the species is very sensitive to these changes, which again goes to show how sensitive they are in the natural environment. All in all, we have in place various conservation practices, being the natural habitats being protected, reintegrating the species from lab grown seedlings. However, there's clearly limitations to these practices, that being the highly specialized microhabitats that's required for the species, the dependence on specific mycorrhizae fungi. If the fungi is not present in the natural habitats when it's being reintroduced, it will not succeed. A high mortality rate during acclimatization, as mentioned before. The host tree specificity. It is best survival on pop ash and pineapple. Substitutes reduce the establishment's success, and this goes back to preserving and protecting the natural habitat. The species is slow to grow and has a low reproduction output, so a lot of conservation efforts are being and resources are being put into producing and protecting the species and reintegrating them with very low uh, value in return, right? They're not a lot of those individuals are going to survive, which then brings into question whether or not it's worth funding this research, right? Um, Pathogen vulnerability is another problem. The exposed roots are prone to fungal and bacterial infection during acclimatization, and in fact, throughout the entire protocol, if they're being grown in a lab, right? If that's why sterilization is highly important. If there's any ounce of contamination, you have to restart from the beginning, which then goes back to the economic uh, resources that are being put into the conservation practices. And then, of course, the poaching pressures still remain, which is where laws are in place and institutions have begun um, finding ways to limit, right, the illegal poaching of the species from its natural habitat. For example, Fairchild uh, offers and sells the species that they grow in the lab themselves, and hopes that people will divert from poaching them if they're already available in the market through the labs. Thank you for watching, and this is my list of references. Have a good day.